Hello, Dr. Sheila, am I audible?
Yes, sir. Yes, sir. Yes, sir. Your voice is audible, sir.
Okay,
Yes, sir. Most welcome you.
thank you.
JP, JP, sir.
Good evening, sir. Hearty welcome, Ashok, sir.
Good evening,
Happy
Dr.
to see
Jayapal,
you.
sir. Hello, thank you so much,
Hello.
sir. Nice meeting you, sir.
Yes. Hearty welcome.
Seems like Tirupatur weather is very cold.
Very cold. Not only Tirupati, everywhere it is cold.
That's true.
Extreme cold, extreme hot.
Yes, sir.
Yes.
Okay.
I think some 20 participants have joined. Okay, we shall
Okay,
wait
okay.
for
Let,
another 5
let others join.
to 10 minutes. We'll start it.
I'll wait.
Thank you.
Good evening, Ashok.
Good evening, Yas. Good evening, Yas.
How are you? How are you,
I'm
man?
good. I'm good. How are you? Nice to see you.
Thank you, na. Thank you.
Fernandez Anna is also there, it seems, huh?
அசோக்
Anna, how are you?
எப்படி நல்லா இருக்கிறியா? எப்படி சென்னை மலை?
மழை இப்போ ஒன்றும் இல்லைண்ணா. That is better. மழை ஒன்றும் இல்லை. நார்மலா தான் இருக்கு. மழை
Okay,
சீசன்
okay. புது ஒன்று வருதா எனக்கு வருதா?
இப்போ ஒன்றும் தெரியலண்ணா. ஆனா வெதர் ரொம்ப ஒரு மாதிரி ஹீட்டியா தான் இருக்கு. இன்னும் ஒரு நாளைக்கு அப்புறம் இன்னைக்குதான் வெயில் வந்தது.
இதை போட்டுக்கிறாங்க திரும்ப கடந்த ஒரு அப்படி போட்டு வச்சுக்கிறாங்க. சென்னை எப்படி போட்டு வச்சுக்கிறாங்க.
ரொம்ப டல்லா தான் இருக்கு.
For this opportunity.
Hearty welcome to the outside participants. We shall start this FDP within 5 minutes. Let us wait for other participants to join. Welcome you all on the behalf of PG and Research Department of Biochemistry. Clear. Screen is visible, Ashok sir.
It's perfectly visible, sir.
Perfect, perfect.
Thank you. Thank you.
Sheila ma'am?
Yes, sir.
Okay, ma'am. Just check. We'll start sharply at 6:30.
Sure, sir. Sure, sir.
Yeah, sir. Everything is fine?
Yes, sir, everything is fine, sir.
Okay, sir. Thank you, sir.
Can you hear me?
Yes,
Yes,
sir.
sir.
Yes,
It's
sir.
clear. Ashok, sir, shall we start? I think it's 6:30.
Yes, as you wish. You can start.
Sheila ma'am?
Yes, sir. Yes, sir. Yes, sir.
Yes, kindly proceed, ma'am.
Yeah. Thank you, sir. We have waited so long to have you among us, and at last the time has come. Very good evening, one and all present here. On behalf of Sacred Heart College, cordially welcome you all for the 5 days online faculty development program on global scenario and future prospects in life sciences, organized by PG and Research Department of Biochemistry, Sacred Heart College. Welcome you everyone. Prayer should be the key of the day and the lock of the night. Now I invite Dr. PG and PhD head, Dr. A. Jayaprakash sir, to lead the prayer service. Please, sir.
Thank you, ma'am. Am I audible?
Yes, sir.
Okay. So I welcome all the participants. Let us all seek the blessings of God. So kindly you can close your eyes and we will start the prayer service. Wishing all a very warm as well as love-filled evening. As we gather here to reflect, to discuss, to share, to acquire knowledge, we take a moment to place ourselves in the presence of our Lord. Living God, you call us to be in the hands and feet of Lord in our world. Stand us now in your presence that we might be refreshed and renewed. Guide us. The God's plan, every single person is called upon to grow. Each life is a vacation from birth. Each one of us carries within ourselves the seed of personal growth. Each one of us can bear the fruit proposed for us by God. Lord, help us to work together and make this FFDB program meaningful. We may fan the flame of God's light in our path and enable us all to the hands and feet of the Lord. We make this prayer throughout our Lord. Amen.
Thank you, sir. To be inspired is great, but to be an inspiration is an honor. A warm welcome to all of you. At the very outset, with a sense of deep respect and gratitude, I would like to welcome our rector and secretary and principal to this FDP. A hearty welcome to dear fathers. It's a great honor and pride in welcoming our resource person of today's FDP session, Dr. Ashok Kumar, Associate Professor, School of Life Sciences, B.S. Abdul Rahman, President, Institute of Science and Technology, Chennai. Thank you, sir. Thank you for your accepting our invitation, for your great support and being with us to share your knowledge, sir. Hearty welcome you, sir. With immense pleasure, I would like to welcome Dr. A. Jayaprakash, sir, PG and PhD head, Dr. M. Fernandez Durai, sir, UG 8 for this FDP. whose support is the key for this program. Welcome you, sirs. I extend my warm welcome to our Mrs. J. Reshma, UG Association President, dear colleagues and research scholar from our institution. Welcome you all. And also I welcome to all the heads of the department, faculty of our institution, and valuable participants of professor, research scholar from other institution. Welcome you all. Optimism is the faith that leads to achievement. It's the golden rule, the simple idea that if you treat people well, the way you would like to be treated, they will do the same. Now I request our dynamic PG and PhD head, Dr. A. Jayaprakash sir, to give away the inaugural address and overview of 5 days FDP. Please, sir.
Thank you, ma'am. So good evening to one and all present here. It is indeed a great honor as well as privilege to inaugurate the 5 days online FDP program on global scenario and future prospects in life sciences, organized by the PG and Research Department of Biochemistry, Saidot College, Autonomous, Tirupattur. At the outset, I extend my warmest greetings to the esteemed resource persons, moderators, faculty members, and participants joining us from various institutions around India. The theme of this FDP is both timely and significant. In a world increasingly shaped by advancements in life sciences, our ability to harness knowledge, research, and innovation is pivotal for addressing the challenges of this 21st century. From improving human health and combating the disease to developing sustainable environmental solutions, life sciences are at the core of its progress. This FDP program has been meticulously designed to offer insights into cutting-edge topics such as tissue engineering, regenerative medicine, molecular advances in gynecological malignancy, microbial breakthroughs, and the role of nature in addressing health conditions like ulcerative colitis. The inclusion of a session on funding opportunities and strategies for entrepreneurship and skill development highlights the practical aspects of leveraging our scientific knowledge for societal health. The FDP will also serve as a platform for collaboration, networking, and exchange of ideas among researchers and academicians. It is through such innovatives that our strengthen expertise, inspire innovation, and pave the way for impactful contributions of life sciences domain. I extend my heartfelt thanks to the organizing secretaries Dr. K. Sheila and Mrs. J. Reshma and all the faculty members of the PG and Research Department of Biochemistry for their relentless efforts in curating such a meaningful program. I am also grateful to our distinguished resource persons who have graciously agreed to share their expertise and experience with us. With this, I declare this FDP on global scenario and future prospects in life sciences officially inaugurated. I am confident that these 5 days will be intellectually enriching and will motivate all to continue striving for excellence in our respective fields. Wishing you all a fruitful as well as engaging learning experience. Have a nice day. Thank you.
Thank you, sir. Now I would like to call Dr. Hai Niyaz sir, Assistant Professor, Department of Biochemistry, to introduce our resource person. Please, sir.
Dr. Sheila?
Yes, sir. Yes, sir. Your voice
Am I-
is audible.
am I
Sir.
audible?
Yes, sir.
Yeah, thank you.
Yes, sir.
So-
Yes, sir.
I wholeheartedly thank our conveners of this 5 days FDP program, as well as organizing secretary for giving me this opportunity. I feel happy to introduce Dr. Ashok K. Pandurangan, who is my senior while I'm studying in Manimbudi Islamia College. Dr. Ashok K. Pandurangan is a distinguished associate professor at the School of Life Sciences, B.S. Abdurrahman Crescent University, Chennai, with a PhD in biochemistry from the University of Madras. Dr. Ashok's research expertise spans oncology, toxicology, and phytopharmacology, emphasizing signaling pathways and cancer prevention. His academic and professional highlights include: he is the top 2% scientist listed by Stanford University in 2020, 2021, as well as 2022 in oncology and toxicology. His awards and recognition include: he is the recipient of the C. Raman Prize in the year 2022. Best PhD Thesis Award in the year of 2011, University of Madras, and the Best Faculty Award from the Crescent University in the year 2021. His editorial roles include Associate Editor at Frontiers in Cellular and Infection Microbiology and the Consulting Editor for journals like Drug Development and Therapy. His leadership includes Vice President of the Society of Phytopharmacology and Research in India and Faculty Coordinator for the Association of Cancer Education and Research. His research contribution include over 82 peer-reviewed publications with a total impact factor of 211 and 3,377 citations. It includes the H index of 32, I-10 index of 50. He authored 28 book chapters and delivered more than 25 invited lectures globally. His lead and collaborated multiple funded projects, including international research grants. Mentorship of Dr. Ashok has guided numerous students, including 3 PhD scholars, 31 MSc students, and several undergraduate researchers. Fostering the next generation of scientists. His professional memberships include he is an active member of esteemed scientific societies, including the Indian Association of Cancer Research, Nutrition Society of India, and the Society for Biotechnologies India. Dr. Ashok's vast academic experience, exceptional research contributions, and leadership in scientific communities makes him an invaluable resource person for our faculty development program. With this short bio, I would like to invite Dr. Ashok Kumar for his update. Welcome, sir. Can you proceed now, sir? Thank you, sir.
Thank you so much, Niyaz, for your detailed introduction. First of all, I would like to thank organizers, especially the convener of this particular FDP, Dr. Jai Prakash. And also, I would like to thank the organizing secretaries, especially Dr. Sheila and Mrs. Reshma. I know as a teacher how much pressure we used to undergo with academic load. Apart from that, it was always a pleasure to organize such events so that we can meet and discuss and share our knowledges. So thank you for setting up a wonderful platform to share our knowledge and meet the other faculty members of different colleges and universities. I hope this kind of events will be, I mean, your department will organize more so that we can share and meet each other. I also would like to thank the management for encouraging the faculty members to conduct this kind of events. Seems like Sagadat College is like almost like my college because most of the people are there, my seniors and my juniors. So I feel like I go to a kind of Islamia College and University of Madras. So I always never said no if any kind of invitation comes from Sagadat College. So I always love to come. Even my hometown is Tirupattur. People, I mean, I would like to share. So it's always homecoming to Sagadat. Unfortunately, I can't able to study there, but I'm happy to associate with Sagadat College through this kind of events. So now I'd like to go into my topic, harnessing the nature of remedies for ulcerative colitis. So this particular study, which I started I started learn from my US postdoctoral study, where I started work on ulcerative colitis and colitis associated cancer. I've been introduced there, My PhD topic is completely different from this because I worked in colorectal cancer where I developed natural product mediated drugs for colorectal cancer prevention. So this is a quite new kind of study where we learn, where I learned from my postdoc boss. So from there I continued, still I continue this in my studies. So so this is a small story about this building. This is the building where you see the background is School of Life Sciences. We have 7-story building dedicated to only life sciences research. Life sciences research and teaching as well. So that is a small introduction about this building. Before I go into ulcerative colitis, I would like to give an overview about inflammatory bowel disease. So generally other participants, please mute. Inflammatory bowel disease is generally defined as there is an inflammation in the colon. So it is further classified into ulcerative colitis and Crohn's disease. So what is the difference between ulcerative colitis and Crohn's disease means? The inflammation that occurs throughout the colon means that is termed as ulcerative colitis. When this inflammation that happens in patches, that is not in continuously in the colon, that is called as Crohn's disease. So both are inflammation, but the difference is only whether it is a patch or throughout the colon is affected. So if the throat colon is affected, that is called UC. So generally UC is nothing but inflammation in the colon, I told you already. So though the etiology is unknown, but generally it is believed that the environmental and genetic and immunological factors that contribute for the development of ulcerative colitis. So what is the clinical manifestation of this particular disease means? Generally the patients will undergo severe abdominal pain. They may feel diarrhea, and some patients may feel mucus or bloody stool. This is the main major symptoms that occurs in terms of ulcerative colitis. But what will happen at the molecular level means generally the oxidative stress is the main culprit to initiate this particular disease, where the constant activation of oxidative stress that leads to the chain of events that activates the inflammation, where the inflammatory cells which is triggered by this oxidative stress, which leads to the recruitment of pro-inflammatory cytokines such as TNF-alpha, interleukin-1 beta, interleukin-6, 17, and 21. So these particular mediators were reach to the inflamed sites and which try to cause severe inflammation in the colon. Due to this severe inflammation, there is severe damage that will affect in the villi of the colon. So coming to the model that is supposed to develop for ulcerative colitis, I hope you researchers, you already know that generally the model which you need to study that need to be very precise, then only you can able to develop any sort of drugs. So for ulcerative colitis, there is one classical model is there, that is dextrin sodium sulfate induced model. So dextrin sodium sulfate is nothing but which is a polysaccharide, where the molecular level of 36,000 to 50,000 kilodalton. If you use this particular molecular weight, this will cause a severe inflammation in the colon that will exactly mimic like ulcerative colitis. So I have given the structure of this is the structure of dextrin sodium sulfate. So when you give this particular dextrin sodium sulfate to the rat or mouse, so generally we prefer to use a mouse model. If you give a 3.5% of dextrin sodium sulfate in drinking water for 7 days, which causes acute inflammation. If you want to continue for chronic inflammation, you should give 14 days a break, and then again you give a second cycle, and then again 14 days break, and then you give third cycle with a 7 days of 3.5% of dextrin sodium sulfate in drinking water, which cause chronic inflammation. So if you want to induce acute inflammation, the 7 days of 3.5% DSS will be good enough to cause inflammation in the colon. On 7th day, you will see the inflammation. So what is happening in what kind of molecular changes that happen in the ulcerative colitis, especially when you give dextran sodium sulfate? What will happen is generally this neutrophil, the inflammatory cells, neutrophils, eosinophils, and macrophages, they are all upregulated in the initial cycle. That leads to the activation of TNF alpha, interleukin 12, the VCAM, and interleukin 17. During the recovery phase, these particular symptoms or these particular molecular events that will come down. Again, when you give a second cycle of DSS, even this particular signaling molecules are aggravated, which leads to recruitment of more inflammatory cells. Even at the chronic level, you can see the CD4 T cells also will be released. So this is what the scenario that will happen during the introduction of dextran sodium sulfate and recovery phase. So why we need to taking very serious about ulcerative colitis means, if the untreated ulcerative colitis can cause a cancer, which is very, very specific, that is called colitis associated cancer. This is the colitis associated cancer is generally termed as a cancer that arises in the colon, but that is totally different from the sporadic model of cancer. This is generally inflammation driven cancer model. So we need to be very serious. We need to treat this ulcerative colitis at the early phase to avoid the formation of colitis associated cancer. So generally, inflammation is always troublesome. Even people, you might heard that even autoimmune diseases are really, really troublesome for people to treat. So this is a kind of autoimmune disease. This is a kind of autoimmune, but it is not autoimmune where the inflammation is higher. So generally, people will prefer strong antioxidants can be a better option. So we in our lab, we were really interested to study the natural antioxidants, especially flavonoids or the compounds which is derived from plant materials or medicinal plants. So why we choose plant material? I mean, plant-derived drugs means they are quite safe and they have already proven it has potential antioxidant activity. So it is always preferable to use this particular kind of naturally derived plant molecules. So in this study, we have chosen three type of molecules, flavonoid and sulfur containing natural compound. A flavonoid is gallic acid, and sulfur containing compound, which is halicin, which is derived from garlic. And the boldin is a type of alkaloid which can be isolated from Numus boldus tree. So this particular talk is going to see the effect of these three molecules, how they try to protect the colon from the exposure of dextran sodium sulfate. So the experimental setup is generally group 1, which is normal control, group 2, which is different percentage of dextran sodium sulfate. I have mentioned 2.5 to 3.5% because when you purchase a dextran sodium sulfate from different companies, they vary, their effect varies. Some dextran sodium sulfate, let's say if you buy from MP Biochemicals, this particular dextran sodium sulfate is very, very good at 3.5% dose. But if you buy from Alpha as a company, the dextran sodium sulfate 2.5%, it is very effective. So that is why I have mentioned to 2.5 to 3.5%. And the third one is the DSS group treated with natural product for 7 days. So this study will be totally carries on 8 days. On the 8th day, we'll do the sacrifice. So what are the parameters we use to analyze means disease activity index? Colon, we measure the colon length and we measure the spleen weight and we study the histopathological analysis. And we also check the immune cells recruitment, what is the status of immune cell recruitment. And we also check the expression of inflammatory mediators, how these natural products that controls the inflammatory mediators. In the first study, we chosen gallic acid at 10 milligram per kg body weight of gallic acid, which is treated to the animal orally for 7 days. So in this study, we initially checked the body weight of the animal. In the control animal looks normal, and the dextran sodium sulfate induced animal showed there is a substantial reduction in the body weight. You see the red line. This is purely because the animal is losing its body weight. That is because their colon is completely damaged. They cannot able to absorb any nutrition. So that is why within the 7 days they started lose. I mean, on the 3rd day onwards they started lose body weight. When you give the gallic acid at 10 milligram per kg body weight, there is a substantial improvement in their body weight. So this gave us a little clue that okay, some gallic acid is protecting the colon at some extent. So we would like to go further to study further. On day 8, we sacrifice the animal and we measure the colon length. You check the in this graph we have shown the length of the colon. So we cut the colon and we measure the colon length from the anus to the rectum. Generally, why this colon is shrinks in the dextran sodium sulfate induced animal means the dextran sodium sulfate has the ability to erase the villi. You see the colon, there is a brush border structure of villi is there. So the dextran sodium sulfate has a tendency to erase this structure because of there is no architecture there. So the colon gets shrink. That is why the length is shortened. So when we treated with the gallic acid, the length was substantially improved. That shows the gallic acid is protective. And we also measure the spleen weight. Why we need to measure the spleen weight means generally the spleen will compensate the blood loss by producing more blood cells. So in such case, in dextran sodium sulfate model, in the figure D, you see the dextran sodium sulfate, the spleen weight is substantially enlarged. But when you treat it with gallic acid, the size of the spleen was less. That is because there is less blood loss that happens during the treatment. So after we have certain promising results, we would like to go further and check the microscopic observation of colon. So we check the histopathological analysis. You see the A is control and C is dextran sodium sulfate induced model. When you see the structure of colon in the control, you see the brush border structure, which is not really affected by anything because we haven't treated anything. So the brush border structure is very nice and the architecture is good. And when you check the dextran sodium sulfate induced model, there is complete destruction of brush border structure. There is no proper differentiation of villi is there. So this is a typical picture where the dextran sodium sulfate has affected the colon. And when you treat it with gallic acid at 10 milligram per kg body weight, the regeneration or some kind of protection that is there in the colon. The same things were observed in the scanning electron microscope also. When you see the 2.5% the control colon, the surface of the control colon, there is no such damages happened. But when you see the 2.5% treatment colon, where you see the structure of the surface of the colon is completely damaged. And when you see the 2.5% DSS model treated with gallic acid, you see that the damage is less. That means these areas are completely protected. But when you see this image, the whole area was affected. There is no proper structure when you compare to the control. You see the villi, very nicely seen the villi. So the microscopic observation also, microscopic observation also given us a better clue. So some kind of protection is happening. So we would like to study at the molecular level. So we check the gene expression pattern of these pro-inflammatory cytokines, especially TNF-alpha, interleukin-1 beta, interleukin-6 and interleukin-17, and also interferon gamma. They are all upregulated during the dextran sodium sulfate induced model. You see all of them were upregulated. So this is the kind of clue that the inflammation is higher in these DSS induced animals. When we treat it with gallic acid, there is 2-fold or 3-fold reduction in the gene expression pattern in the RT-PCR analysis we have checked. So generally gallic acid offers protection by controlling the pro-inflammatory cytokines recruitment in the inflamed colon. We also checked the inflammatory cells, status of inflammatory cells. So we checked two markers, one is CD68 and another one is MPO. CD68 is nothing but which is cell surface marker, which is expressed in neutrophils. And MPO is nothing but it's a myeloperoxidase, which is an enzyme, which is act as a marker of monocyte or macrophage. That is a differentiated monocyte is differentiated into macrophage. So this particular expressions or activities will give us the clue what is the status of these inflammatory cells that is present in the colon. So when we check the immunofluorescent analysis, you see the dextrin sodium sulfate, these regions is red color dye, which is CD68 positive, which is merged with the Hodges dye. But when you treat it with gallic acid, there is no such expressions are observed. And similarly, when we check with myeloperoxidase, where in the dextrin sodium sulfate model, the myeloperoxidase, as expected, the myeloperoxidase activity is high. That is because of the macrophages are highly accumulated in the inflamed colon. But when you treat it with gallic acid, the MPO activity was substantially reduced. So this is another positive indication where the inflammatory cells recruitment is very low in the colon. And then we check the expression of NF-kappa B. You know very well NF-kappa B is a central regulator of inflammation, which is a transcription factor. And we check the expression of NF-kappa B and their expression in immunohistochemistry and Western blot. When you see the dextrin sodium sulfate induced model, the expression of NF-kappa B is substantially high. That was shown in the graph also. We quantified through ImageJ software. And when we treat it with gallic acid, the expression of NF-kappa B is less, and their counterpart inhibitor kappa B was high in the gallic acid treatment. The similar result was observed in the immunohistochemistry, immunofluorescence analysis also. So this shows gallic acid controls the inflammation by inhibiting the NF-kappa B, via it is controlling the recruitment of pro-inflammatory cytokines in the colon. Finally, we would like to study a signaling pathway where it connects the inflammation and tumorigenesis. So especially this pathway is very crucial. People might know already. This is a niche between the inflammation and cancer. See, especially this interleukin-6, which is a pro-inflammatory cytokine, we all know. So this acts as a ligand for this JAK-STAT pathway. When it binds to the JAKs and JAKs get phosphorylated, the phosphorylated JAKs in turn activates the STAT3. STAT3, when it gets phosphorylated, it translocated into nucleus, where it activates genes which is responsible for cell proliferation. When you control interleukin-6, you can control the whole pathway. You can prevent the formation of tumor. So we would like to check the expression of what is the status of gallic acid and STAT3 signaling. So when we checked this phosphostat3 in the DSS group, the levels were comparatively substantially high. When we check the total STAT3, there is no such difference is happening. Similarly, STAT3's downstream target is BCL-XL. So in BCL-XL is also higher in the DSS model. When we treated with gallic acid, you see the substantial reduction. The similar results were observed in the immunofluorescence also. So this overall study gave us a clue that gallic acid can control the inflammation by inhibiting the neutrophils and macrophages recruitment in the colon. That controls the pro-inflammatory cytokines in the colon. That further controls the formation of tumor where the interleukin-6 is inhibited and there is no further activation of STAT3 path. So we managed to publish this paper in the International Immunopharmacology. Next compound we have chosen as halicin. Allicin, you know already, allicin is one of the organosulfur compound which is isolated from garlic. So the garlic smell, you see the strong odor. That is because of this allicin which is present in the garlic. So allicin, we treated 25 milligram per kg body weight of allicin to the animal. Again, the experimental model is also the same, where we have 3 groups. We have found we check the body weight. When we check the body weight, as expected, dextran sodium sulfate treated animals, they've drastically gone down within the day 2 status. When we treated the animal with allicin, which gives us the better result than the gallic acid. The similar results were observed in colon length and spleen weight also. We have found a substantial increase in the colon length when we treated with allicin. This is the microscopic observation, where you see the colon is completely damaged, 2.5% DSS treatment. But when you treat it with allicin, you see the colon is somewhat protected from the exposure of dextran sodium sulfate. When we check the histological score, also we have seen the similar graph. Similarly, we check the status of immune cells recruitment in the colon. When we check the immune cells recruitment, especially through the CD68 expression and MPO activity, we have found that the 2.5% DSS exposure, the immune cells, especially the CD68, is positive in the dextran sodium sulfate. But when you treat it with allicin, the expression was substantially reduced. Similar results were observed in MPO also. These 2 results indicated that allicin controls the recruitment of both neutrophils and macrophages in the colon. Then we checked the expression of pro-inflammatory cytokines. Here we could able to get only four pro-inflammatory cytokines, especially TNF-alpha, interleukin-1 beta, interleukin-6, and interleukin-17. All these 4 pro-inflammatory cytokines levels were higher in the dextran sodium sulfate used to model. When you treat it with allicin, they are substantially reduced. This gave us a clue that allicin has a tendency to control the inflammation. When we checked the NF-kappa B status as a central regulator of inflammation, we found higher expression of NF-kappa B in the dextran sodium sulfate induced model. The activation of NF-kappa B is the main reason for the inflammation, aggravation of inflammation in the colon. But when you treat it with allicin, the NF-kappa B levels were substantially reduced. The similar results were shown in the quantification. And as I mentioned in the previous studies, when we checked the status of phospho-STAT3, we found higher activation of STAT3. And when you treated with allicin, the phosphorylated form of STAT3, that is an activated form of STAT3, where their levels were substantially reduced. So we managed to publish this particular paper in the Oxidative Medicine and Cellular Longevity. And the third one is the Boldin, which is Boldin is an alkaloid. So in this particular study, we studied the disease activity index. So disease activity index is nothing but this is how the scoring system is done, where we check from the day 1, we check the weight loss percentage, stool consistency, and whether the blood is visible in the feces. So each of this condition have certain scoring. For example, if an animal loses its body weight 6 to 10%, initially the animal's body weight is 30 grams. So it loses body weight about 3 grams. Let's say it is 27 grams. That means it lost 3 grams, that is 10%. That means we give a score 2. And also if the stool consistency is loose, generally the stool consistency is very tight. in the normal animal. If it is loose, we give 2. And if it is slightly bleeding, we give 2. If the animal have only 5% of reduction, we give 1. Similarly, we score each day to each animal. Then we collate all this we sum up all these scores, and finally we plot a graph. When we check the disease activity index, when the control animal doesn't show any kind of any sign of disease, and when we treat it with disease, the dextran sodium sulfate, you see the extreme activation of disease activity. This means the dextran sodium sulfate is causing some kind of damage in the colon. So when you treat it with Boldin, you see these signs were drastically come down. So this shows the Boldin is offering some kind of protection. When we check the spleen weight, where the spleen weight is higher in the dextran sodium sulfate induced model, when you treat it with Boldin, their levels were substantially reduced. The similar results were observed in the colon. The shrinkage of colon is very high in the dextran sodium sulfate model, but when you treat it with Boldin, their levels were substantially increased. So this is the macroscopic observation that also shows the similar results. In terms of microscopic observation through H&E stain, you see the damage of colon completely. This is a normal villi. This is completely erased colon. You see completely these structures are destroyed. You see the normal colon villi, this looks like this, perfect. But when you treat it with Boldin, we see there almost look like a control animal. But so some kind of patchy damages are there. Also, some kind of inflammatory cells recruitment is also. These dyes where you have similar, the recruitment of neutrophils and macrophages are happening. That shows some kind of inflammation. And then we check the expression of CD68 through conventional immunohistochemistry. And instead of immunofluorescence, we check this time conventional immunohistochemistry. These brown patches, they are all indication of CD68 expression. But control, you look, there is no such expressions are there. In the Boldin treated also, there is few areas you can find, but it is not as such seen in the dextran sodium sulfate model. So similar results were observed in the MPO activity, where the MPO levels, the activity is higher, but when you treated with the Boldin, their levels were significantly reduced. So this is an indication where Boldin is controlling the inflammatory cells recruitment in the colon. In this model, we could able to get only positive results on 3 cytokines, especially the expression of TNF alpha, interleukin 6 and interleukin 17. All 3 cytokines genes were upregulated. But treated with Boldin, that gives a very, very promising result. You could see only there is almost a 6-fold activation of all those genes, maybe 3 to 6 folds. But when you treat it with Boldin, it's almost, you look like control. Among these 3 compounds, I felt like Boldin is very, very effective. The similar results were obtained for NF-kappa B, which I don't want to explain further. It may be boring to you. And to further, we checked this expression through in vitro model also. So we took the raw 264.7 cell, that is a macrophage cell line. We activate this particular cell line through LPS 10 nanogram. And then we isolated nucleus fraction and cytosolic fraction, and we checked the expression of NF-kappa B. And in the nucleus fraction, we have found higher activation of NF-kappa B. When dose dependently, we treated with Boldin at 2 different doses. We found a substantial reduction of the NF-kappa B expression in the raw cells. So similar expression were observed in the immunofluorescence also. Again, when we check with the expression of phosphostat-3, the expression is very, very higher in dextran sodium sulfate induced model. But when you treated with Boldin, this particular level was completely, substantially reduced. And similar results were observed in the immunohistochemistry also. So what is the overall mechanism that these three drugs can induce means? So when you treated with the dextrin sodium sulfate, which activates NF alpha and it activates NF kappa B, where in turn it activates the downstream pro-inflammatory cytokine, that is interleukin 6. The activation of interleukin 6, which activates further JAK pathway, JAK-STAT pathway, where when the phosphorylated STAT3, which will enter into nucleus, and where it activates the genes which is responsible for cell proliferation. But in terms of treatment with allicin and gallic acid and boldin, where it inhibits directly NF kappa B, it inhibits TNF alpha, and it also inhibits the phosphorylation, that is the translocation of STAT3 from the cytoplasm to the nucleus. So this is one of the key findings that we found so far, where it controls inflammation as well as tumorigenesis. So we managed to publish this paper in Biofactors. So all these three works were published. These are all the list of publications in this field that we publish. And I hope I completed my study. If you give me 2 more minutes, I will acknowledge some of my bosses so that hope they will be happy. So this is my first postdoc lab where I worked in California, the Children's Hospital Research Center. Here I learned this particular disease model, that is ulcerative colitis. She is my boss, Dr. Julie Saba, and we had a very big group. This is my first postdoc and the second postdoc. All of these studies were done in this lab. She is Dr. Noraizan Mohammad Esa from University of Putra, Malaysia. She is my second postdoc boss, and it was a very small lab. So we were in a conference. Here I did all these studies. And this is my third lab where I worked with Professor Mohammad Rais Mustafa, where as a visiting research fellow, he is my boss. So we had a very big group in University Malaya. University Malaya is very famous university in Malaysia. That is the #1 university in Malaysia, and which is also in the top 100 ranking there. So this is my research team. I have 6 scholars. Unfortunately, 1 scholar joined late. So she's Saranya, Yasmin, Pratibha, and myself, is Kashif and Ashwati. This is my small research team. This is my contact information. Thank you. If you have any questions, I'm happy to answer.
Thank you, sir. Thank you, sir.
Thank you, Ashok sir.
Hurry.
Thank you very much. It was really a wonderful presentation, and your research insights on ulcerative colitis. Really, it was amazing.
Thank you, sir. Thank you.
Sheila ma'am, if the participants,
Sir.
they have posted any questions, you can directly go ahead and ask the questions.
Yes,
Or
sir.
they can
Yes, sir.
feel free and they can unmute and they can ask directly to the resource person. We have another 15 minutes time.
Yes. Dear participants, if there are any questions, kindly unmute and
Not
directly ask
ask,
the
they can
questions
put
to
it in
you.
chat box also.
Directly ask the
They can.
They can notice. They can type in the chat box also.
Yes, yes, yes.
So in the meantime, just I have a question, sir, Ashok sir.
Yes, yes, please go ahead.
So you've done a lot of research on this ulcerative colitis.
Mm-hmm.
So one question that is related to the diet. Should the ulcerative colitis patient should change their diet? Is there any connection?
Definitely, it impacts the diet. So unfortunately, I didn't present a study. So this high cholesterol diet, we have done a parallel study in US itself. So we have given an animal with a higher amount of cholesterol, high fat diet, generally high fat. So we taken two different diets. So which have one particular diet is in very normal, and the second diet is which is high fat content. People who eat beef, especially high fat, cheese, those who eat pizza, that same calorie level fat. So those animals within 7 days to 14 days, we found that the inflammation is very high in the colon. The overall inflammation is very high. So this particular disease is highly connected to diet. And people who have high amount of fat intake, they can able to get high chance of this particular disease.
Okay, sir. Thank you. Thank you. Can this UC can heal on its own?
It is no. So far, UC cannot see. Generally, in the animal treatment, when you expose to DSS model, it can heal. But as per the disease that comes in the patient, it won't heal. So main problem of this particular disease is since it is inflammation, it is body's necessary action. So you cannot able to give any medicine. Generally, so far people used to treat the non-steroidal anti-inflammatory drugs. That is the only medicine so far available. So when you take so much the higher amount of NACIDs, what is the side effects you will get. So this is another part of the study. So there is no permanent solution is so far is there for ulcerative colitis. Unfortunately, my student also developed a scholar, one of the scholar have ulcerative colitis. She is also going the same kind of agony. She cannot able to get proper medication. Doctors used to treat only the non-steroidal anti-inflammatory drugs. When you get non-steroidal anti-inflammatory drugs, they have their own side effects. So which will damage your liver. So once your liver is damaged, your metabolism will be affected. It's all chain of events that occur.
Thank you for answering in detail.
Thank you.
I think there are some questions in the chat box. Sheila ma'am?
Sir, Ashok sir. Yes, sir. Yes, sir. Sir,
You can go ahead with one by one.
Yes, sir. Yes, sir.
What is a permanent solution for? Yes.
Yes, sir. Okay, sir.
What is a permanent solution for ulcerative colitis? I don't know who is the name of SHC-Bio. Okay. So the permanent solution so far, no. So far, there is no permanent solution is there. Only doctors are prescribing non-steroidal inflammatory drugs. So the side by side, this kind of natural products, the clinical trials are going on. So far for curcumin, they have done the clinical trial. They have gone for curcumin, but it is no such promising result were observed. Still, the research is very seriously going on in this field to get a better drug. So far, whatever we have done, it's all preclinical only now. So we should need more and more research in this particular field to have better drug. Harita Prabhu has asked a question. What is the interplay between interleukin 6, JAK-STAT, and TNF alpha NF kappa B pathways, and how do natural agents disturb their culture? I think I have given an overview about this pathway. So generally, NF kappa B is a central regulator of inflammation. So where it connects with all the pro-inflammatory cytokines, it activates pro-inflammatory cytokines on one side. So generally, this happens a kind of paracrine and autocrine signaling. So paracrine and autocrine signaling is generally this particular inflammation has happened between epithelial cells, CD4 cells, neutrophils, macrophages, and with this. So what is autocrine and paracrine? Let's say if one immune cells get initiated with inflammation, so which will produce the pro-inflammatory cytokines, TNF alpha, interleukin 6. This will activate its own cell when it is released from the cell, and which also goes and activate the other cells, such as, let's say, macrophage, which produces releases TNF alpha and interleukin 6. This macrophage, this cells, I mean, this signals which is released from macrophage will also go and bind to neutrophil. And it will also go and bind to epithelial. So which in turn it activates its own cell and other cells. That is why it is called as autocrine and paracrine. So this is the inflammation link. So there is a niche between inflammation and colon is called interleukin 6 and JAK signaling. So interleukin 6 is a pro-inflammatory cytokine which is known already, which is released in a Th1 subset. So interleukin 6, which is released from the any type of cells, let's say if it is from the macrophages or neutrophils, which can go and bind to this JAKs pathway in the epithelial cells. When it binds to the JAKs, which activates this STAT3. STAT3 is one of the important regulator of cell signaling, which has a tendency to activate more than say 60-70 genes, which is responsible for the cell proliferation. All leads to cell proliferation. You know what will happen when the switch is on? It's continuously on. When the cell proliferation is higher, the switch is on, that will end up in cancer. This is what the kind of inflammation and tumorigenesis link. How this natural product disrupt means, generally these natural products, I haven't shown some of the results in this papers. Generally, these particular natural products, they are highly antioxidant. If you control control the production of reactive oxygen, that is the top regulator of inflammation. ROS, inflammation, and cell proliferation. This is the hierarchy. When you hit the head, that is the ROS. When the ROS is controlled, then automatically the inflammation will be controlled and the tumorigenesis also will be controlled. So most of these particular drugs, they act as a potent antioxidant. They can control they scavenge the free radicals and thereby they restore the cell's original status. This is how they regulate. This is the one mechanism they regulate. And the other mechanism is through NRF2 signaling, where I have shown in the other paper, the gallic acid does potentially activates NRF2 signaling. NRF2 is nothing but which is also a kind of transcription factor, where it has a tendency to activate enzymatic antioxidants in the body, in the cells. So high amount of NRF2 activation leads to high amount of these antioxidants, enzymatic antioxidants such as SOD, catalase, GPX, GR, all get activated. Those enzymatic antioxidants will scavenge the free radical and they restore the cells. So this is how either directly this natural agents scavenge the free radicals, or sometimes through this NRF2 mechanism, which will be taken care. This is how they regulate.
Thank you for a clear explanation, sir. I think we can take the final
Harita
question.
asked one question. Harita asked one question. These natural agents were able to focus on programmed cell death. Yes, when you give, see, the dose is the major issue for a cell or a particular cell undergoes apoptosis or offers protection. A natural agent, when you treated the cancer cell, that act as a pro-oxidant, sometimes it acts as an antioxidant. Dose is a matter. So these same kind of drugs, when you treat for cancers, they act as a pro-oxidant. They induce cell death, programmed cell death in case of apoptosis. That happens, yes. What are the other organs that affected by colitis? And generally, so far, the major organ that is affected by UC is colon. Colon is the major target. A small portion of intestine is also get affected. That is the ileum part. The ileum part means where the colon starts, intestine ends. That part is called ileum part. So that part is get affected by ulcerative colitis. So yes, intestine is also affected.
Thank you, sir.
Okay, one more question is there.
Vera ma'am,
Yes, sir. Yes, sir.
One more question is
sir, want
there.
to
Is
take
there-
that questions? Some 2 or 3 questions are there in chat box.
Yes, sir. Yes, sir.
Yeah. Is there any other pathway involved in this other than JAK-STAT pathway? Other pathways are there, especially this cell addition molecule. The activation of cell addition molecule is higher in C model. That pathway is also there. So my motto is to control the tumorigenesis, control the inflammation and control the tumorigenesis. That's why I study in this pathway. But other pathways also I studied caffeic acid phenylethyl ester CAPE I have used for treatment of ulcerative colitis. So there I studied ICAM and VCAM expression. So that particular CAPE has given better results. Yes, that pathway is also there. So the main pathway JAK-STAT is, which is involved in inflammation and cell proliferation. That's why I used to study this part.
Thank you, sir. I think it is the right platform to say that Dr. Ashok, he has been listed as 2% top scientist in India in the field of cancer biology. Congratulations, sir. Congratulations for your wonderful research
so
and
much.
ulcerative
Thank you so
colitis.
much. Thank you. I think the last
Happy to
question I
have.
would answer, why you are chosen only these 3 bioactives only? So I would like to study more bioactives. Now my research students are working on, now we have over with land plants. So our focus is now shifted to marine-based natural products. So we started that particular area also. So that particular time we are interested in this particular drugs. There is no specific reason we chosen these drugs. So I think I answered your question, Dr. Pungodi. There is no specific rationale there to use these 3, but generally we have checked with cocoa also, cocoa polyphenols also we checked because the Malaysian cocoa was not explored that much. So our lab that particular time we studied what is the effect of cocoa diet on colitis associated cancer. So cocoa eating chocolate controls inflammation and tumor. So I can suggest you can have a chocolate. That's good for your health.
The last one more question, sir.
So diagnosis, they asked if it
It's
is
a
diagnosed young age. Okay, thank you. So this ulcerative colitis, it is not gender specific or age specific. This can come to anybody. Okay. Only thing is we are not aware of this particular disease because it is very, very common in Western world. In US, if you go to Europe, it is very common. But in India, this is very alarming now. That is because of we used to have very healthy diet previously. Now we are also stick to the Western diet. We eat high amount of fat in our diet nowadays. We are addicted to pizza, burger, and other high fatty foods. So cheese is not our diet. It's not our regular diet. Nowadays, people are very addicted to this kind of junk foods. That is the main reason. And the oil also, the fried oils, which we are not very sure what kind of oil they are using. That is also most of the oil which you procure from the companies, they are all saturated because once it is saturated, then only it will last long. If you have unsaturated fatty acid is there, that will spoil. You will get the smell odor. That is why companies they convert all these unsaturated into saturated by the process called hydrogenation. So that is the main reason the oil lasts long. Even 1 year also the oil will be there. So better we avoid high fat diet. So stay away from this disease. That is the only message, take home message I can give you.
So, sir. So thank you,
Yeah.
sir.
Thank you so much, ma'am. Thank you.
Yes, thank you, sir. Really,
Anybody
most of the
would like-
participants
Yes.
He
if you
explained it well,
like to
as well as very
contact, I am giving
informative
my-
knowledge.
Thank you,
I'll
Ashok
Yes, sir.
sir.
Your
Wonderful
contact
insights.
details also I will share the
I'll
the
put
group.
it in the chat box so they can email me if they need anything they want to talk. Thank you
Sheila,
so
ma'am,
much.
Yes, sir. Yes, sir. Dear
we're going
dear
to conclude, ma'am.
participants. Yes, sir. Yes, sir. Dear participants, feedback link will be posted in the chat box. Kindly fill it. It will be activate only few hours. Dear participants, the feedback link will be posted in the chat box. Kindly fill it. It will be activate in few hours only. Participants, thank you all for your active participants. Participants are kindly requested to stay connected for tomorrow's session at the same time, 6:30. Thank you all.
Thank you.
Shogana,
Thank you.
last
Thank you.
slide,
Thank you, Dr.
Shogana.
Jayaprakash.
Sir, yes, sir.
Shogana.
Ashok, Niyas is in line. Niyas, sir?
Yes,
Yes.
yes.
Last slide, last 48th slide, please.
48th slide. Okay.
What happened?
Yes, due to curiosity, I want to see.
I could able to share. I'm
Such a
not able to
wonderful session, sir. Wonderful session on
I
ulcerative
can't able to
colitis.
share. Can
Really,
you
it
allow
was
me
a
to share?
fruitful
Can you able to see
evening.
my slides?
Yes, it is in 46th.
Oh, 1 minute.
46 like
48.
that.
Okay. That is my
They
BTech
are
group.
all.
They are my BTech group. They are all BTech project students. So that is right.
Fine. Thank you. Okay. Thank you.
Yeah, thank
Thanks
you.
for your availability. Thank you.
So one last information. This is the picture of Golden Gate Bridge, which connects Sausalito and San Francisco. So hope it's one of the iconic bridge which was built more than 600 years, I guess. Not exactly. So where in one cold morning, so Alcatraz Island, you people heard, it is one of the highly, highly guarded prison. Hope you heard about Alcatraz Island. That is there on my left side. So this is a backwater which comes from Bay, which goes inside up to Berkeley. This is the picture where I took it from my past postdoc. Thank you.
Thank you, Ashok sir, for your wonderful insights.
Thank you so much.
Looking forward for your valuable present in future.
Thank you,
MPP or other
Dr.
programs.
Jayaprakash. Thank you, all of you. Thank you, Sheila ma'am.
Thank you all. Have a nice day. Thank you.
Thank you, sir.
Thank you, sir. Thank you so much.
Thank you, Anna. Thank you for your availability.
Thank you, Ashok.
Thank you, Niyat. Thank you.